Dear students, today we will discuss development of ovule in angiosperm. So now let's discuss the pre-fertilization events in case of the angiosperm, especially the female gametophyte. So first of all, we will look at certain terms which are important to understand and clarify. The female reproductive part of the flower is called pistil, which is also known as gynoecium, and when it composed of one carpel, it is called monocarpellary. When it consists of two carpel, it is known as bicarpellary, and three, then it is known as tricarpellary. So carpel is the unit of pistil. Next is ovule, which is known as megasporangium, and it bears the embryo sac. Embryo sac is known as female gametophyte or megaspore. The egg cell, which is also known as oosphere or female gamete, and fertilization, which is also known as syngamy. Now let's look at the pistil, which is known as the female reproductive part of the flower. So it consists of three parts: the stigma, style, and ovary. The upper portion is the stigma, and the basal one is ovary, and the ovary bears the ovules. Okay, and these ovules are arranged in a certain way in the chambers. These chambers are called locule. So again, depending on if the ovary has one locule, it is known as unilocular. If it has two, it is known as bilocular. If three, then trilocular, and then for many multiloculars. Now let's see what is ovule and what is the structure of ovule and how the female gamete occurs in ovule. So ovule represent inside of ovary and bear the embryo sac. The ovule is attached to the wall of ovary with a mass of parenchyma cells called placenta. So here you can see this structure is called ovule, and then you can see it is present inside the ovary. Okay, and then space is there which is called locule. This space is locules, and then it is attached with these cells placenta, and then the ovule is not directly attached to placenta but connected by the stalk called funicle. So you can see the stalk, and the point of ovule that is attached to funicle is called hilum. So this is the same hilum which is present in the seed. So it is the point of attachment, and that inside the ovule you will see the embryo sac. Now here we will see the development of ovule. Now initially ovule consists of mass of parenchyma cells which is known as nucellus. So this we are talking about the developmental stage. Okay, so nucellus functions to provide the nourishment. Then multicellular coat develop around the nucellus, and this is called integument. So first the mass of cells is called nucellus, and after that the outer layer they develop integuments, and they protect the tissue of ovule. The point from where the integument arise is called chalaza. So here in the diagram you can see these are the mass of cells called nucellus, and after that the layer which develop is called integument. This could be single layer or bilayer. Okay, and then you will see the point where the integument arise is called chalaza. So from here they are arising, this is called chalaza, and this end is called chalaza end, and the opposite end where the ends there is a pore. Okay, and this end is called micropylar end, and this is a pore which is known as micropyle. Okay, so this is what is how the ovule is formed. The integument can be single or bilayer. They grow from the base to top. As the top, the small opening is left in the integument, and this is called micropyle. So this provide another, you can say, opposite polarity to the developing ovule. So we can say ovule has two polarity. one is its chalazal end, and one is its micropylar end. So usually the pollen tube enters through the micropylar aperture in most of the cases, but not in all the cases. So now we will see the development of female gametophyte, that is the embryo sac. So at micropylar end, the hypodermal cells differentiate to archisporial cells, and then periclinar division occurs. As a result, outer and inner layers are developed. The outer one forms the parietal cells, and the inner one forms the sporogenous cells. So the sporogenous cells differentiate to megaspore mother cells, and the megaspore mother cell undergo the meiosis and it forms megaspore. So as a result, 4 megaspore are formed. Out of 4, the 3 megaspore they degenerate, and the fourth one it enlarges and forms the embryo sac. So we can say the embryo sac, that is the megaspore, is formed from the megaspore mother cell after the megaspore mother cell undergoes meiosis. Now this megaspore mother cell undergoes series of development to form the embryo sac. So how that occur? The megaspore nucleus it divide, okay, and then it get converted into two nuclear stage after the first mitosis. This is also known as interchangeably, this is also known as two cell stage, but it is basically the division of nucleus which is going on. And then again second mitosis occurs. So out of two, four nucleus are generated. This is called four cell stage of embryo sac. And then third meiosis occur and 8 cells are formed. And then these cells arrange themselves at the polarity. 4 of them move to the micropylar end, and 4 of them move to the chalazal end. Okay, so 4 nuclei move to the micropylar and 4 to the chalical. One from each end move toward the center. Now one move toward the center and fuse to form the secondary nucleus at the center, and later on the cytoplasm develop. So this is called 8 cell stage. Okay, so at micropylar end we have 3 nucleus or cells. Out of 3, one is called egg cell and 2 are synergids. In the center, 2 nuclei fuses and form one polar nuclei, which is 2n. And at chalazal end, 3 nuclei are there, and which are known as antipodal nuclei or cells. So if you look at the diagram of embryo sac, this looks like this. Here you can see the egg cell along with these synergids from the egg apparatus, and here you can see this is micropylar end. These are called filiform apparatus. They have finger like projection and they are rich in polysaccharide. It guide the pollen to the synergids. Then egg is there. Basically the egg cell fuses with the male gamete to form the zygote, and this is polar nuclei, which is 2, and this is also known as central nucleus or cells. This is also known as definitive nucleus. And then 3 antipodal cells are present at the chalazal end, and they possess number of enzymes of starch, lipid, etc. to provide the nourishment. So if you look at the definitive nuclei, this again fuses with another male gamete, okay, and this is only the diploid structure we can say in the embryo sac, and it gives rise to primary endosperm. So if you look at the mature megaspore, okay, or functional megaspore, it is actually so called as 7 cell stage and not the 8 cell stage. The growth of megaspore or gametophyte stop at this stage, and this is called haploid. The overall structure is called haploid structure. The growth is resumed after the fertilization and the development of embryo sac, and the monosporic as only one megaspore is formed during the megasporogenesis, and the process of development of megaspore from formation of megaspore from the megaspore mother cell is called megasporogenesis. Okay, so again in short, if we revise that, the female reproductive part has the ovary, so which consists of ovule. And if you look at the ovule, consists of three parts: integument, the outer layer, nucellus, the inner cell mass for the nourishment, and then we have embryo sac. So how the embryo sac is formed? It is formed from the megaspore, and megaspore formed from the megaspore mother cell, and these are formed from the sporogenous cells. So here you can see the embryo sac. One function is that it produces egg cell. Another function is that it forms the molecular basis of fertilization, because fertilization occurs here, and later on the seed development also occurs in the embryo sac, where the embryo sac get converted, not the whole one, but part of it is converted into embryo. So this is all about the development of ovule. Thank you.